Coming up on Dr. Kiki Science Hour, we're talking about emerging diseases with Tracey Goldstein and Suzan Murray from the PREDICT Project. That's up next on Dr. Kiki Science Hour.
Netcasts you love.
From people you trust.
This is TWiT.
Bandwidth for Dr. Kiki's Science Hour is provided by Cachefly at CACHEFLY.com.
This is Dr. Kiki's Science Hour with me, Dr. Kiki, episode 132, recorded Thursday, February 9th, 2012, Playing Catch. This episode of Dr. Kiki's Science Hour is brought to you by Netflix. Watch thousands of TV episodes and movies on your PC, Mac, iPad, iPhone, or TV instantly. All streamed directly to you, saving you time, money, and hassle. For your free 30-day trial, go to netflix.com/twit. Welcome, everyone, to Dr. Kiki's Science Hour. I'm Dr. Kiki Sanford, and this is episode 132 of the series. We have spoken with many, many scientists over the last couple of years, and today is no different. We're going to be talking about science. We're going to be talking about animals. So you know how it goes. One topic, 1 hour, one expert. But, oh, wait, this week it's two experts. Why two? Well, you know, why have rules if you can't break them occasionally? So I'm breaking my own rule and having a couple of experts to talk about a really interesting subject. I hope you're ready to get really deep into the subject of emerging diseases and tracking emerging diseases through the new PREDICT Project with our guests Suzan Murray from the Smithsonian National Zoo and Tracey Goldstein from UC Davis. But first, a few science headlines. It's February 9th, 2012, and this is the science that makes headlines this week. Astronomers from UC Santa Cruz and Carnegie Institution found an Earth-like planet just 22 light-years away. The planet, named GJ667cc, is rocky, estimated at approximately 4.5 times the mass of the Earth, and exists within its red dwarf star's habitable zone, even though it takes only 28 days to complete its orbit. Interesting side note is that GJ's star system has 3 stars. Oh, and this planet was actually discovered last year, but this study confirms that fact. Russian scientists at Vostok Station in Antarctica report breaking through the 4,000 kilometers of ice that has sealed Lake Vostok away from the rest of the planet for the past 20 million years. Scientists around the world hope to discover microbial and other life forms that might have evolved in isolation down there. Debates rage concerning the probability of contaminating the lake with life from the surface. Svante Pabo and his team at the Max Planck Institute for Evolutionary Biology opened the full Denisovan genome database for analysis and comparison by other scientists. The genome itself and related conclusions will be published by Pabo's team in coming months. Open science for the win! We will certainly be hearing more about the information gleaned about humanity from a tiny finger bone in the coming months. Results of the latest LHC analyses of the CMS and ATLAS experiments were published and increase the Higgs boson signal to approximately 4.3. This suggests odds of over 99% that the Higgs exists. But, you know, as scientists, they're still not certain yet. So for more certainty, we will have to wait until the LHC starts running again this spring, possibly at even higher power. This is the week you will learn the best glass for serving champagne. Well, maybe not the best, but the possibilities that you could use. According to a study in PLOS ONE, French researchers found that slim champagne flutes allow a greater collection of carbon dioxide gas than broader coupes. And colder champagne releases less ethanol, so pour accordingly. Teeny tiny tarsiers were found to hear very high-pitched sound, as high as 91 kilohertz. To confirm that the tarsiers themselves could make the sounds, researchers recorded the animals using a bat detector. It's still unknown how the small creatures make the noises, but it's thought that the sounds sometimes serve as alarm calls and possibly in detecting insect prey. The Indiana Senate passed a bill allowing the discussion of various religious origin stories in the public classrooms of the state. The intention of the bill's sponsor, Dennis Cruz, was to insert the teaching of creationism into science classrooms alongside the theory of evolution. However, the Speaker of the Indiana House made statements suggesting that the bill would not be brought for a vote. New research funded by the Bill and Melinda Gates Foundation suggests that malaria may kill nearly twice as many people worldwide as previously estimated. Published in the journal The Lancet, the study concluded that deaths from malaria were 1.24 million in 2010 rather than 655,000. On the bright side, the numbers of deaths do seem to be decreasing year to year. And that does it for the science headlines this week. Let me know what you think about the science news, or tell me what you think should be news by emailing Dr. Kiki at drkiki.tv, or leave me a voicemail at 650-741-5454. Now for a word from our sponsor. This episode of Dr. Kiki Science Hour is brought to you by Netflix. Netflix streams thousands of TV episodes and movies directly to you instantly, which means that you save time, money, and hassle, which is great, right? We love saving hassle. There are several easy ways to instantly access streaming movies and TV shows with Netflix. You can watch Netflix TV shows and movies on your Mac, your PC, your iPad, if you have an iPad app, or even your iPhone and some Android phones as well. If you have a gaming console like an Xbox 360, a PS3, or a Nintendo Wii, you can watch Netflix right on your television. If you're not a gamer, but you have one of those cheap and handy, small set-top boxes, you can watch Netflix on your TV with an Apple TV or a Roku box. With Netflix, you can watch movies and TV shows instantly using any of these devices, and you can begin watching a movie or a show on one device and then finish on a different one. Whichever way you choose to access Netflix, you can watch as many movies and TV shows as you want, anytime you want, and you can cancel anytime. So try Netflix today for 30 days, free. Just go to netflix.com/twit. Be sure to use this URL when you sign up for your free trial. It's Netflix, netflix.com/twit, TWiT. We thank Netflix for their support of TWiT, and we hope that you enjoy watching instantly with Netflix. Now it's time to get into our show. Let's start talking to our experts. Today we're talking with Tracey Goldstein from UC Davis. She is a research faculty with the Wildlife Health Center and Laboratory and the surveillance coordinator for the PREDICT project. And with Suzan Murray, she is the chief veterinarian at the Smithsonian National Museum. Ladies, thank you very much for joining me today.
Yes.
It's great to have both of you on. Tracey, let's start with you. I was wondering if you could give us an overview of what the PREDICT project is.
Yeah, so this is a new initiative from the U.S. Agency for International Development, or USAID, and it's an extension of their avian influenza project, and they name it the Emerging Pandemic Threats Program. And we are one arm of that whole project. There are other arms that have equally interesting names, such as Identify, which are the international organizations such as World Health Organization and Food and Agriculture Organization. Respond, which is primarily a consortium of universities. And then Prevent, which is a group of individuals that do behavior change. And PREDICT's main job is to actually develop surveillance systems in order to identify the next pandemic, so the next HIV or SARS, in wildlife before they emerge into humans.
That's got to be an incredibly difficult job, and I'm really interested in talking with you about that today. How did you get involved in the project?
Well, I think, so first I wanted to back up one step and say that PREDICT is formed by a consortium that is led by UC Davis, but also includes Smithsonian. Obviously, Suzanne's on the phone today as well. And then EcoHealth Alliance and Wildlife Conservation Society on the East Coast, as well as Global Viral Forecasting, which is based in San Francisco. And all of us have been doing this type of work, studying diseases in wildlife in different ways. And so it's something that's really close to our heart and the work that we do anyway. And so the fit was just really a nice one for us to come together as a group and then start to work together on this project. Yeah. And probably the main impetus for it is I'm not sure if most people are aware, but the majority of emerging diseases are actually zoonotic or diseases that come from animals. And within that, 75% of those actually come from wildlife. And so it's a part of the U.S. government's work that hasn't actually been focused on before. There's been a lot of work done on domestic animals and a lot of work done on humans, but really no one until this project had been doing wildlife. And so it's really sort of a progressive idea for USA to want to take this on.
Yeah, and how did you personally get involved, just through your involvement at UC Davis at the Wildlife Health Center?
I have done this sort of work on and off primarily with marine mammals until this project came along. But I did my PhD actually with John Mazette, who is the principal investigator for the project, and then went away and did a couple of postdocs and eventually came back to UC Davis, partly to help to develop the Wildlife Health Center's diagnostic lab to test samples for wildlife. And so when this project came along, I just felt really lucky to be able to be a part of it.
Yeah. Suzanne, how did you get involved, and how did the Smithsonian Institution or the National Zoo get involved in this program?
Well, first of all, we are a sub-awardee of UC Davis, and Smithsonian has researchers in the field all over the globe that conduct research on animals and do a number of different things, from tracking elephants in Sri Lanka to investigating maned wolf health in Bolivia. And so we have a number of researchers that are already looking at wildlife movements. As veterinarians and pathologists at the National Zoo, we use our resources to support and to help co-investigate some of these health and disease issues. So it's something that we've been doing for a long time with both captive animals here at the zoo, but then with wildlife globally. So it was also a very natural fit for us to come together with our partners.
Yeah, and personally, how did you get involved with this specific project?
Well, as the head veterinarian, we have many different projects that are ongoing, and our collaborators are people that I've worked with for years, from GVFI, WCS, and certainly UC Davis. There's people we've all known, and we've partnered on projects together before, so it was a natural outgrowing of previous relationships.
In working together and managing the different relationships that you have, how is this program set up? Where do you even start putting something like this together?
You want to start with that, Tracey?
Sure, then you can just jump in.
Sure.
So, as Suzanne mentioned, many of us have been doing this type of work around the world for many years, and so the way we set it up is each organization leads in different countries, and we generally lead in the countries where we already have projects set up. So UC Davis has a long-standing project in Tanzania, so it made sense that we would be the lead there. And by doing it that way, it allowed all of us to start the project that much quicker. And then within the organizations, each objective is led by a different organization, but we have teams that sit on each one of them. So, for example, the laboratory diagnostic work is being led out of UC Davis, but we have somebody from GVF and somebody from EcoHealth Alliance that sits on the team as well, and we meet all the time, and we make decisions and make the work plan together as a team. And so that way everybody sort of has a role in sort of shaping and changing how we're doing our work. Did you want to add anything to that, Suzanne?
No, I think that's a great introduction.
So let's talk a little bit about how you actually start surveillance for the monitoring of emerging diseases. I mean, when the public hears about emerging diseases, it's after they're already, after it's out. after people have already started getting sick, after it's already made it into a human population. So let's take it back scientifically and can we start at the beginning of the process?
Sure. So I'm not sure if you've seen the paper, the Nature paper by Kate Jones, where sort of the big outcome of that was looking at where diseases have emerged since sort of not really the beginning of time, but really over the last 200 years. And from that, taking a look at population density and mammal biodiversity and producing these maps that we now sort of refer to as the hotspot maps. And those sort of led or highlighted parts of the globe or parts of the world that were high-risk areas for pathogens to have already emerged prior to, the recent years. And these include sort of hotspot areas along the tropics, as you might imagine, in the Amazon and in Congo and in Southeast Asia. So that was sort of the big start. I think USAID saw that paper, and they were really interested in expanding the avian influenza work, and that is where we started from. In terms of operationally, so I'll give you the example of Uganda. So UC Davis is the lead in Uganda, and our implementing partner in the country is the Mountain Gorilla One Health Project, and you may be familiar with them. They're the ones that have managed and helped to mitigate some of the problems in mountain gorillas for many years in those parks. And so because of that, we already had a team on the ground that was working there. Our team are Ugandans, they're not Americans, and they're very well connected with the Wildlife Authority, the Department of Health, and the Ministry of Agriculture there. And so that allowed us to really have a team on the ground doing this sort of work with one species so that we could then expand that to other work that we're doing. And that's a very similar case to what's happening in all of our countries. EcoHealth Alliance has been very involved with that work, for example, in much of Southeast Asia, and they have a team already in the ground, well integrated with the ministries. So that's giving us the opportunity to expand that beyond whatever species we started with in that country.
So really, what I'm finding really interesting about this is using a lot of what's already pretty much infrastructure that's in place, but just laying on top of the groundwork that's already been done.
Exactly. And, given that it's funded by USAID, it's very much a development project. The idea is that we help to develop or further train the teams, develop the laboratory diagnostic systems in country that eventually, hopefully, get taken over by the governments and embraced by the work there. So essentially, putting ourselves out of work. The idea is that the countries at the end of whether it's this funding cycle or if the project get funded again, that the countries are able to do their own surveillance and their own diagnostics.
Yeah, that makes sense. Now, Suzan, in terms of people on the ground who are the veterinarians who might be coming in contact with various animals and actually noticing that there's something that might be emerging. That's actually what got me interested in this in the first place, was a press release from the Smithsonian about a veterinary training course that was going to be taking place. How do you teach them? I mean, veterinarians are already, pathologists in a sense, but how do you teach them what to look for, and specifically this could be something that could be pandemic causing?
That's a really good question. And we have, as Tracey has mentioned, that we've got teams in many different spots around the globe that are trained and run different sorts of programs. In this case, this was a pathology training program. So we had pathologists with us as well as clinical veterinarians. And one of the reasons that the National Zoo became involved in this aspect of the training is that it's an area of expertise. We have a great pathology staff, and so we wanted to make sure we were using that expertise and those resources to help these important goals. In this situation, when we first started trying to look and be prepared to figure out what could be abnormal, one of the most important things is to have, for people on the ground, to have a very good grasp of, well, what is normal? Mm-hmm. So we weren't really sure before we have our teammates that work in Vietnam and live in Vietnam. So we had some idea of of what the training should be. So we together with Hanoi University and WCS and UC Davis and Smithsonian and as well as a pathologist from University of Illinois. We spent a lot of time preparing, trying to figure out, well, at what level should we start? And we wanted to make sure that we pick some of the major different species and make sure that we start out with our trainers to teach and to ensure that they knew what was normal in a bird, like a chicken, and what was normal in a necropsy of a pig and a frog. So we could then from that teach, well, what might be abnormal, what should you watch for, and what should you do if you see this.
Right. So go from normal to the actual pathologies, the problems. And what do you teach then? So what happens after that if a veterinarian or if someone who comes in contact on one of these teams with something that's not normal?
Well, let me just back up one step too, because the whole idea of what's normal, anatomy changes so much from species to species. So we started out, we had chickens, and what we had found, we had wonderful, wonderful colleagues from Vietnam, Laos, and Cambodia, but their training was different in veterinary school than the training that we have here. And the majority of our participants had never had the ability to, or the opportunity to conduct a necropsy on their own chicken. They've watched other people do it, but they haven't done it themselves. And there's a huge, huge difference between watching someone else doing it and then doing it yourself.
Right.
And the same thing with a rabbit and a pig, and even simple things like making blood smears. So you can look at that under the microscope and look at those cells and how many of this type and how many of that type and what's normal. It was such an exhilarating experience because our colleagues, they were so happy and so interested. So we felt like we were making a huge impact just even on the first day by sharing the knowledge of what we had.
Yeah, and in terms of, you mentioned that the idea of normal is different for different species. You've got a chicken, but a chicken is not a parrot, is not another type of bird. So is there a wide range of stuff that they might be missing out on just by having these kind of stereotypes that they're studying with?
Yes, there's so many different species of birds, right? So you're right. A parrot would be much different than a rat type and different than a chicken. But we also had to start somewhere. And, so we started with the basics. And, but again, felt we made a lot of progress. And not only examining the bird or the rabbit or the pig, but note-taking. So describe what's normal and describing what's abnormal. And then after that, we taught sample collection as well. So during that, how you collect samples, and very importantly, how do you do it safely? How do you do it safely for people? And one of the most important things that we do is, first do no harm. So while we're trying to teach it, and use our resources to help educate about what could be abnormal, we want to make sure we're not putting anyone in harm's way. So we spend a lot of time on health and safety measures as well.
Is everything considered abnormal until proven normal as one of the safety measures?
Yeah, I would say that it's a good way of looking at it. We spent a lot of time with personal protective gear and masks and Tyvek suits, and so yes, that was one of the points we tried to get across. Until you're sure that it's not an infectious disease, treat it as though it was. So yes, that's one of the big principles of personal protective gear.
And in terms of the surveillance, where we hear a lot here that there's the animal trade at markets in some of these highly populated areas, and that's where many diseases will come across the animal to human barrier. Who do veterinarians go out and just search? Are they scouting for possible species vectors? How does that work?
I'll take the first stab at this, and then maybe turn over to Tracey because she's going to have some good thoughts on this as well. And interestingly enough, the type of things that could happen in a market market might vary from country to country or continent to continent. So in Africa, in the hotspots in Africa, for instance, you might have more bushmeat, and so animals that are not alive but that anymore, but meat that was taken from an infected animal in a forest that could be more of a danger. While in Southeast Asia, it's typically live animals that will come into contact with different species in a market setting where in the wild they never would have. So you could see a monkey with a bird next to a fish and a human. So the risk factors in Southeast Asia are different than they are in Africa or other areas. So yes, so having said that, the strategy for trying to identify and protect and prevent does indeed vary with what you think the risk factors are. So it wasn't our team per se, but we do have the broader Predict team that is working in markets in Asia and Africa as well. Doing surveillance like that, many different types of surveillance. So surveillance by working with animals in the wild and collecting biological samples. Surveillance in the markets, and actually the pathology workshop is a form of surveillance as well, because we wanted to make sure that our partners would have an idea of what to do if they came across 50 dead bats. Is that an issue? Is it not? How do you investigate it safely for yourself and for everyone else as well? So it's a different sort of form of surveillance. Yeah. Tracey, what did I miss on that? Do you want to add in?
Yeah, no, I think that was a great explanation. What I wanted to do was sort of take a slight step back, and we started with talking about the hotspots. So that helped to focus what countries we were going to be doing some work in. But then from there, we had to target our surveillance strategy even further, because we couldn't work everywhere in every country, and we also can't work on every species in every country. So we had to take a second look and try to come up with what we now call a SMART surveillance strategy, which is a targeted now I'm just forgetting the word. Targeted and responsive, basically, surveillance strategy. So what that means is we targeted our species to focus on 3 primary host taxa, and these are primates, bats, and rodents. And we chose those because primates are most closely related to humans, so of course that makes sense. Also, when you look in the literature, we have a lot of shared pathogens between primates and humans. And then bats and rodents, again, if you look in the literature, are the next groups of animals that also share a lot of pathogens with humans. And that's because they live in such close contact, especially in these developing places where rodents are, often sharing the same food sources as the people, etc. And then the third is what Suzan was getting at, and that is looking at these, what we were calling them interfaces. So there's places where humans and wildlife come together. And as Suzan mentioned, those really vary by country. So in Asia, a huge area where this occurs are in the markets and also in the wildlife trade. But often if you go to other places such as South America, that's often occurring through, land use change and deforestation. So we're trying to target our work at those places where humans are coming in most contact with these animals, and where the potential for transmission of diseases is the highest.
Yeah. What happens when a disease is recognized by somebody in the field? What is the procedure that you have in place?
So there's a couple of things. First, a lot of what we do is actually sampling free-ranging, potentially healthy animals. And that's because oftentimes wildlife they may be the host of the pathogen, but it doesn't always make them sick. So when they spill over from wildlife into, say, domestic animals or humans, that's when you start to see disease. So probably about 95% of our work is focusing on free-ranging animals at these different interfaces. But then the other 5% is sort of what you are talking about, where either there's an outbreak of humans, and then we go and do sampling of animals in the area to try to see if we can find sick animals, or sick animals that we may find that are having a die of themselves. And then we do the sampling that way. And so we have protocols that we've developed and we've used to train our teams all around the world. And they have an idea of, first of all, how to protect themselves, as Suzan mentioned, what sort of protection they should be wearing when they're going out and sampling these animals, as well as what types of samples to collect, how to collect them, how to store them, how to handle them. And then in the cases of human outbreaks, if we're invited by the governments to assist, we will go in and at the government's request, help to sample wildlife and other domestic animals during the outbreaks. Suzan, anything you want to add to that?
No, I thought it was perfect.
I know that you're talking about, you keep mentioning that there are multiple hotspots in countries around the world, and so different teams are going to possibly have different government regulations, different methodologies to doing things. How are you globally coordinating your efforts so that you can, when you do find something in a country, actually let the rest of the world know? We predict that this is going to be a dangerous area, or that something is going to possibly come out of here.
Yeah, so in terms of our teams are all trained with the same protocols and the same types of training. And then they, in turn, are actually helping to train the ministry officials and the veterinarians and the epidemiologists in those countries. So we're working really closely with the governments. In many of the countries, the labs where we're doing the testing are actually the government labs or closely associated or affiliated with the government labs. So we're working really closely with them so that they know what we're testing and what we're finding while we're finding it. And then once we find something that either we're worrying about immediately or think that needs some follow-up, we're working with those governments to do that follow-up work so that they're sort of a part of that every step of the way. So essentially, we don't want any surprises. Mm-hmm. And then, the sort of the process for approval of release of data varies, as you mentioned, by country. Some are much more concerned about others. I also think, finding a herpes virus is probably less concerning than finding a new Ebola virus. And so those sorts of circumstances will change as well. But we are not able to release the data to the general public until the government gives us approval to do that. Of course, the exception to that is when you have some really highly pathogenic virus, say a new Ebola, and the government's really sort of reluctant to help to release those data. Then in those situations, the international regulations sort of step in, and those warnings have to be put out, and those actions have to be taken to protect human life. Yeah.
Suzan, did you have anything that you wanted to add?
No, not yet. Sorry,
not yet.
Suzanne,
you were mentioning the dealing with government organizations. So at a certain point, you have to be communicating with the CDC, and if in the United States, for example, or whatever the health organization for the given country is. How does this work with more global disease possibilities? So avian flu is thought to be spread as a result of migratory avian species who they travel thousands and thousands of miles around the world between countries. And this is going to be very different from, say, a rodent species that just hangs out in Southeast Asia.
Yeah, so first of all, the way we're doing our testing is we're calling it sort of the family or the genus level testing. So what we're doing is, rather than testing for a specific virus, so say avian flu, we're actually taking it up a level and testing for orthomyxoviruses. So that's the family that flu belongs to. And so what that allows us to do is to detect the normal, or maybe not normal, but the known stuff. So we would detect avian influenza, but we'd also detect anything new. And any of the diseases that are regulated, such as avian influenza, foot and mouth disease, classical swine fever, etc., there are already regulations and reporting avenues that have to be taken if you find those types of viruses in animals. And so we would follow those regulations no matter what. So the way we're setting it up is so that we can sort of find those as well as the new ones. And then obviously, with the new viruses that we may find, obviously there's no reporting system yet. There's no surveillance system in there. And so our hope is to be able to characterize them. And if we think that they're important enough to be included into the surveillance system, then work with the governments to have those incorporated. Also work with the international organizations to have those added to those reportable diseases. And as I mentioned in the beginning, those organizations are also a part of this project, and CDC is a part of that as well. So we have quarterly meetings where all the groups come together, and we have cooperation from those organizations as well, so that when we do find something new, that we can get those incorporated into the regulations and the global surveillance systems.
Yeah, so that it goes up the chain accordingly.
Exactly.
In terms of Suzan, in terms of the veterinary perspective on these diseases, how do we approach here in the United States how we look at, say, flocks of birds versus other countries?
Oh, that's a good one. And I guess it also depends whether there's an current outbreak or not, if there's something we're concerned about. And that's also an issue just if with animals in captivity at the National Zoo. If there's an avian influenza outbreak, how do we protect our animals and the people? And if we're concerned about birds moving in or out. So I would say that within the United States, there are a number of different organizations that look at different bird species as well, both migratory and non-migratory birds that do health and disease examinations. Like even at National Zoo, one of our partners here in Predict is the Pete Mara from the Smithsonian Migratory Bird Center. So he does a lot of health studies on migratory birds as well. So anyways, my point is, even when there's not an outbreak going on, there are a number of different organizations doing surveillance. And then when there is an outbreak or some sort of issue, there's many different groups that can work, use all their resources to work even more closely together to address that particular issue. Like whether it was the Gulf, right? The oil spill in the Gulf, all of a sudden a lot of resources get focused in that area. That wasn't infectious diseases so much, but that was a health crisis in animals where a lot of different organizations came together. So that's sort of how we look at it in the States. And the models in other countries vary a little bit country by country. But Predict's approach to that is somewhat similar. As Tracey had said, we have ongoing surveillance on animals that appear healthy, and then certainly if there's any evidence of a disease outbreak or an issue, then we would focus our resources more and mine down a little bit deeper.
Yeah, that's one thing that I've always thought is just fascinating about the epidemiology of these zoonotic diseases is the fact that they have to come from a population of animals where the majority of the animals appear healthy. They can't just kill off their host immediately because then they wouldn't be as easily transmitted. It just
right, and then wildlife. Right. And then wildlife are also so good, even if they have a disease, at hiding it, right? Because if you show that you're ill in the wild, you're more likely to get picked off. So. Right, so it can be even more difficult to identify, and as we go, whether it's bat or a rodent or a bird, and there are other species that we work with along the fringes, depending on the country and depending on the situation. We, as veterinarians, it's actually a great thrill to be able to use our training so we can safely catch those healthy animals, get the samples, and then release them so we're not doing any harm. So you had asked earlier about the roles of veterinarians, and that's a good example of where we can use our expertise to help animals in the wild, get the samples we need, make sure we don't do any harm, and then help human populations as well. So to some extent, it's a dream come true, the perfect job. We got into this because we wanted to help animals, and now we're helping animals and people. How much better can it get?
That's right. It's double your money.
yeah, right. And it's also this, sort of on a broader level, the last, 5 or 10 years especially, there's a lot of attention paid to sort of the One Health concept and ecosystem health, and more physicians and veterinarians are working together. I mean, we used to be so separate, and now we really work together and combine our expertise to look at health and disease issues. So it's really an exciting time to be in this field.
As far as an exciting time in the whole one world concept or one health concept, I mean, there's a lot of talk about climate change, and especially in how it's going to affect the developing world. But climate change is going to affect the entire world, and we're already seeing different species migrate to new areas. We're seeing whole ecosystems changing as a result of climatic changes that are occurring. As far as predictability, how can you start? Is it more and more challenging? I guess is the question here.
I'll start with that, and then Tracey, why don't you jump in? But I would say that it is very challenging, but that's why we have a whole bunch of modelers on our team, and people who can look, that are really smart, who can look at this and figure it out and give us an idea. But certainly, as temperatures change and warm, vectors of disease spread. They overwinter at more northern areas. Mosquitoes are not dying off in Canada, so we're seeing over the last few years West Nile virus going into Canada. As ocean temperatures change, the migration patterns of whales, turtles will change. So, yeah, with global warming, there's an awful lot that will change, and our surveillance networks are set up to look for that. And of course, that's where I started with the modelers, who are really key players here.
Exactly. That's what I was going to say as well, is we do have a modeling team that's led by EcoHealth Alliance, but we have members from all of our organizations on that. And that original hotspot model that, as mentioned, the primary drivers were population density and mammal diversity. We're now including other drivers such as land use change, climate variability, all those sorts of things, to then try to predict, where diseases might emerge and how it might change, how we should be doing our surveillance. And so the nice thing about this project is we started, with this original model, and then we're continuing to update it. And then ultimately the results that we're producing will go back into the model to change it. So it's a constant circle of trying to improve the type of work that we're doing. So I think it's a different way to do this type of work. But I think because it's so adaptive, hopefully it'll allow us to make those changes as we're seeing the climate's changing and changes in species. And I wanted to just add on something both of you guys said is, I think you, Kirsten, mentioned epidemiology, and you, Suzan, pathology, and that's the thing about this project is, that One Health aspect and putting it all together. And I think when we go into many of these countries, a lot of these international organizations have done a wonderful job of training the labs and the teams to do, for example, surveillance for avian influenza. But it's very sort of focused one type of work. And the difference between us and what we're trying to do is exactly that, is to not just look at one problem, but to say, okay, so now you found the virus. Is there pathology associated with humans? What's happening in the environment? To bring that all together. And I think that's why it is so much fun for us, because we're just seeing the excitement in these countries where they're learning that, once you have a tool, it doesn't have to be used just for avian influenza. It can be expanded to look for other diseases, or, to examine a new situation. And so it's just so exciting to be able to contribute to that.
I think Tracey made that the other point, which is we're working with just such a tremendous group of people, with dedicated people who have dedicated their lives to wildlife health, and with varying expertise. And it really is such an honor to be able to work with such brilliant people with such diverse and expertise, and then for us to be able to knit it all together. And that says a lot to Tracey and Dr. John Mazet, who's the PI on the project, to be able to herd cats, right? To get a very diverse group of people working so well together. It's really just a great honor. Tracey, are you back? I thought I saw you.
I'm back.
Tracy is back.
Yay.
Yay. Okay, now that I have both of you back, what was the last bit of the question
you were
I was
asking
about?
You were asking about the likelihood of
emergence rates. Yeah.
Right. Tracey, that might be more you, huh?
Well, I think you probably have a bunch to say too, so I'll start and then let you take over. I think first, we have just such an amazing and exciting opportunity to, for the first time, be able to even just catalog what these viruses are in wildlife. I think it's really hard to say what's new until we know what's there. So we're setting up our program so that, when you're testing a bat in Vietnam, you're doing the same tests on a bat in Africa. So that ultimately we'll have these new maps of where these viruses are found and in what host species. So I think that's really exciting. But then when we find a new virus, which we've already found more than 120 of them, the next thing we do is we look at their genetics and try to see, is it a bat virus that we found in bats, or is it a human virus that we found in bats? And in fact, we found viruses that go in both directions. We found human viruses in bats, and we found gorilla viruses in humans. So we're already finding evidence for this movement of pathogens between species. And those ones that we find that can infect multiple species are obviously the ones that we want to take a closer look at.
Yeah.
And then we also have to look at as I said earlier, the viral family. So for example, a herpes, it might be something that you're very interested in when it can cross species, because those types of viruses generally don't. But even a herpes may not cause the same type of disease that a new filovirus, so an Ebola type virus might cause. And so we're trying to look at all those sorts of features. And the nice thing right now is that as we're working with these governments, the first viruses that we're finding aren't the big scary ones. And that's helping us to build relationships, show the governments that we can find new viruses and that they're interesting and, things for us to study, so that when we do find the scarier ones, that hopefully we'll have a better trust built up so that we can then jump into that higher level of action that may be needed.
Suzan, did you have anything to add to that? Suzan is gone.
Oh no, we lost her again.
Yeah, well, it makes sense that you want to build those relationships and the trust between the individuals who are working, so that when the big one hits, you can work efficiently and you can work well together, and you've developed that relationship even further. In terms of finding the big one, you don't want to. But how often does something that is considered an Ebola level virus, something that's considered something that's going to be really bad, how often is the likelihood of something like that estimated to be?
That's a really tough thing to estimate, but just based on some of the things right now, just to kind of hazard a guess, I would say 10% or less probably of the viruses that we find. But we have already found new viruses of concern. For example, a new strain of Ebola virus has recently been found by our project that we're trying to understand better how widely it's distributed. And then a new rabies virus. A new rabies virus was found in humans, and obviously that could be quite concerning. So of the 120 new viruses that we've found already probably at least 5 that belong in those families that are of concern. So rabies, SARS-like, Ebola, and then a large proportion of those that have some ability based on their genetics to cross species. And so we definitely are finding already some of those that either need to be put in that box of let's watch them closely, or the box of, okay, let's find out more about them right now.
Yeah. For looking at these diseases, you mentioned that how they're diseases that weren't expected to be able to cross into different species that are crossing into different species. It seems as though the more we're finding about these viruses, it just the barriers between species seem to be things that we as humans have put around them more so than there actually being barriers to crossing over, and that maybe it's just a probability and likelihood issue.
And that's exactly it. I think we don't know. There used to be sort of this dogma of, coronaviruses don't make us sick. Will SARS change that? Or, herpes virus or adenoviruses, maybe herpes isn't a good example, but adenoviruses only affect the host species that they were found in. And now we're finding that that's not true. And so I think the big part of this project is to first gain a better understanding of how these viruses really are behaving before we can take it to that next level of, okay, who are the bad actors and who are the ones we have to really worry about? Because I think we've learned that what we thought we knew 10 years ago, we really don't understand any longer about these viruses.
Yeah, so along the lines of what you're doing in terms of gathering information, it's a lot of learning about things and maybe getting new standards that will be taught to future generations of people who are going to be working on these emerging diseases.
Exactly that, and education of the governments and education of the ministries, because I think everybody's used to thinking in these very siloed ways, and we have to sort of break down those walls and realize that, it's all sort of interactive and affects each other.
Is any of this data that you're collecting going to be opened up? So along the lines of open data, open science, so that
yes,
so it can be shared.
Yeah, exactly. So HealthMap and Google are partners on the project, and ultimately HealthMap will be the public face. So once data has been released by the governments, there will be a sort of a predict page of HealthMap that highlights the countries that we're working in, and the results from our work will be projected on those. So anybody from the public will be able to go to any country and see what species we're testing and what viruses we're finding.
That's just great. Now, I know both of you are very busy women, and you're out in the cold, Tracey, so and the daylight is waning. And Suzan, you have a family to get back to, a dog probably to play with. So I won't keep you much longer, but I would love to thank you for joining me today and for taking the time out of your schedules to talk about this program and help let people know what's going on and what projects like this and the Identify and Respond projects also that are being developed to actually deal with these emerging diseases. I really appreciate it.
It's our pleasure. Absolutely.
Yes. Thank you so much for having us on.
You're welcome. And many thanks to Suzan and Tracey for joining us here on Dr. Kiki Science Hour. For any more information about emerging diseases, you can go to the UC Davis website for the Wildlife Health Center, and you'll get links to the Predict project from there. It's vetmed.ucdavis.edu/WHC. You can also follow WHC UC Davis on Twitter. And additionally, you can always go to the Smithsonian National Zoo website if you want to find out what's going on with our National Zoo. Lots of neat stuff going on. I'm Dr. Kiki, and this has been Dr. Kiki Science Hour. Next week, we're going to be talking about hot matter with a Slack scientist. So I hope you join me next week. Until then, you can follow my sciencey pursuits on Twitter. I'm at Dr. Kiki. drkiki.tv is my website. My Facebook fan page is Dr. Kiki. I'm Kiki Sanford on Google+, and you can subscribe to the show through iTunes, or you can go to twit.tv/Kiki to find past episodes or other ways to subscribe. If you need more sciencey goodness, I don't know what to tell you. The world is full of science. You just, ask questions, follow your nose, and you can find more good stuff out there. I will see you next week. Thanks for tuning into my Science Hour this week. Remember, all I ask for is 1 hour a week, and I do hope that this hour made your world a whole lot more interesting. Now for your science meditation of the week.
The green cell you see here is a killer T cell of the immune system which is attacking the cancerous red and blue cell. You can tell when the killer cell has recognized the cancer cell because the two dots move around and contact the target. The killer cell then spreads out over the cancerous cell. Passing the video through a filter makes the killer cell look yellow and allows us to really see how it focuses on the cancer cell. These killer T cells are constantly hunting down dangerous cells throughout the body and destroying them.